Gram stain and its work. The Gram stain is the most widely employed staining method in microbiology. It's a differential stain because it's divide bacteria into two classes, gram-positive and gram-negative. In the first step of the Gram stain procedure, cells from a fresh culture are transferred from a clean slide and allowed to dry. If the cells are from the agar plate colonies, they first transfer to a liquid medium for a dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from agar medium or from a liquid culture. Fresh cultures must be used because when the cells aged, it's lose their ability to retain stain. The cells are then fixed. The slide bypasses the slide slightly above the Bunsen burner. Fixed cells then stain with the basic dye crystal violet for 30 to 40 seconds. The slide is then rinsed with water to remove the excess stain. At this point, all cells appear purple under the microscope. Next, a solution of grams iodine is added and retained on the slide for about 1 minute. The iodine combines with crystal violet to form diiodine complex to decrease solubility within the cell. At this point, the cells still appear purple. The cells are decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet while the gram-negative bacteria will not. The ethanol or acetone should be added dropwise with the slide in a tilted angle, until the drop goes to the edge of the slide, becomes colorless. Gram-positive cells lose the crystal violet iodine complex during the prolonged excessive decoloration. Excess ethanol is then washed off with water. Under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered by counter-stained safranine for 20 to 30 seconds. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, gram-positive bacteria are purple and gram-negative bacteria are pink. Generally, the gram strainers correlate with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick dehydrated peptidoglycan layer of gram-positive bacteria appears to be permeability barrier and prevent the loss of crystal violet complex. In contrast, the peptidoglycan in gram-negative bacteria is thin and has large pores. Ethanol may extract lipids and increase porosity, thus removing crystal violet iodine complex. If you like this video and want to learn more, please subscribe to this channel and click on the bell icon for updates.